Hi, I'm Mickey Pentecost, co-founder and CEO of Diadem. We make exosome therapeutics. Cells that are touching are in constant communication. But for cells to communicate at a distance, they secrete extracellular vesicles called exosomes. Originally thought to be cellular garbage, exosomes are now a hot new area for therapeutics. Our competitors are focused on what they can put inside of exosomes to deliver inside of cells. We're focused on engineering the outside of exosomes to control cells through their surface receptors. To do this, we engineer the genetic code of immortalized human cells so that the exosomes come out with therapeutically valuable signaling molecules on them. We know their exact size and concentration, and we even know how many proteins we're putting on the surface, ensuring consistent manufacture and dosing. By engineering the surface of exosomes with signaling agonists, we're creating a new therapeutic modality that will eclipse cell therapies and biologics. I say this confidently because of a simple concept we discovered at IndieBio. When we put a signaling protein on the surface of an exosome, it is 10,000 times more potent than the unattached protein. Let's look at this in depth. PD-1 is a co-inhibitory receptor that controls the activity of immune cells. On the right are two purified proteins that bind this receptor. You can see they cause some signaling, but not very much. In contrast, when we put these same molecules on the surface of exosomes, they become very powerful and robust signalers, and they do so at a drastically lower concentration and dose. This is the difference between a research tool and a therapeutically viable platform. Now let me explain why being able to do this is so important. On the left is receptor superclustering, where two cells interact. Since the development of human monoclonal antibodies, pharma has been trying to engineer ones that cause the same stimulatory agonistic signaling. They've gotten really good at blocking signaling, but they can't get this agonistic effect. They can't get the receptor superclustering. What we've engineered here, the ability to do just this, is a game changer for biotechnology. We did all of this at IndieBio, and with the little time we had left, we performed a proof of concept study in animals. We hadn't optimized our dose at all, we were just looking for a signal. Let me walk you through our data. We immunized rats with a retinal peptide, and it results in severe autoimmune inflammation of the eyes. Disease starts at about day 6 and peaks around day 12. Injection into the eyes of saline vehicle or our unmodified exosomes have identical disease progression. In contrast, when we injected our engineered exosomes, it significantly suppressed the progression of disease. As expected, however, there was a half-life and the effect wore off. But a second injection also appeared to have some effect on the rate of resolution. And we saw nearly identical effects when we injected our engineered exosomes intravenously. In addition to our PD-1 agonist exosomes and optimizing other signaling agonists, we're now making multi-specific exosomes with combinations of therapeutically viable signaling molecules on their surface. The potency and versatility allows us to target a multitude of diseases, including cancers, infectious, and neurodegenerative diseases, just to name a few. There are too many for us to target alone, so we're developing assets that we can partner out, and our internal pipeline will focus on inflammatory diseases with unmet needs. So how do we make them and deliver them? We've been hard at work at IndieBio, harvesting, concentrating, purifying exosomes at lab-feasible scales. But we've also worked with a leading bioprocess lab to create a CGMP-compliant, scalable bioprocess for our upstream manufacturing of exosomes from our cell lines. This upstream process uses no animal-derived ancillary materials, ensuring that we can easily isolate human exosomes, and also that we can, for regulatory considerations. In order to produce our exosomes consistently and affordably, we took advantage of the fact that exosomes are the size of viruses. We realized we could apply vaccine manufacturing technologies to their downstream purification. And at IndieBio, we developed our own workflow to separate exosomes from other biomolecules produced by the cells. Importantly, exosomes are more affordable, stable, and dosable than cell therapies, which are also a logistical mess. And because exosomes are remarkably robust entities, we're working on various delivery formats, including inhalable. This opens up opportunities to target rare and common diseases, some with huge markets. We are so confident in our platform and our therapeutic modality that we welcome the biotech industry to come partner with us. We're particularly interested in treating chronic obstructive pulmonary disease. COPD is a growing global problem and the third leading cause of death by disease. It results in nearly $50 billion of direct medical costs per year in the US. And despite having a $14 billion pharma market, there are currently no disease-modifying therapies. We've assembled amazing scientific, clinical, and business advisors to help us reach our goals. Diadem is co-founded by Dr. Wojciech Barkowski, a brilliant biologist and technologist. He and I have been great friends and collaborators for a decade since we were postdocs together in the same lab. I went on to work in biotech, and Wojciech gained expertise in intellectual property as a technical consultant for a leading IP firm. And we've assembled counsel with expertise in building biotech companies and even in prosecuting exosome technologies. And we aim to occupy a large position in this rapidly advancing field. We've mapped our milestones and our use of funds to create tremendous value over the next 2 years. We are now opening up a $5 million seed round to create the therapeutic modality of the future. Please come talk to us. Thank you.